Hello, my name is Jasmine, and today we will be looking at one of the methods that this lab used. The lab that we are focusing on today is the SMAD6 transduces endothelial cell flow responses required for blood vessel homeostasis. The blood vessels include the arteries, veins, and the capillaries. But wait, what does this all mean? Don't worry, me too. After some researching, I found a way to break down this title to y'all. To begin, let's look at the first section. SMAD6 may look a little strange, but for now, we can note that this is a gene that codes for proteins. This experiment then found that this protein uses the endothelial cells to promote blood flow. Here in this image, we see that the endothelial cells are cells that coat the blood vessel. Next, with the production of proper blood flow, we can get blood vessel regulation. So, what's so good about blood flow regulation? Well, regulation is used for transportation of oxygen and nutrients that are needed for the body. And this regulation also allows our body to maintain our body heat. But how do we know that SMAD6 help to regulate blood flow? One method used in this experiment was immunofluorescence. As you can see in the image here, it does a great job providing the visual on parts of the cell. So, how does it work? To start, this antibody is responsible for finding the right antigen. Once it finds the right antigen, then the equipment will read the fluorochrome. So the fluorochrome is responsible for showing the color on the picture. This can be found on top of the antibody. Now that we have gone over the structure of the antibody, let's move over to the image to look at it much closer. For example, we can see here that the green fluorochrome is representing the plasma membrane, and then the pink fluorochrome is representing the cytoplasm, and the blue is representing the nucleus of the cell. Now that we understand immunofluorescence, let's move on to the lab pictures. Here we have the immunofluorescent figure representing the HUVEC, also known as the human umbilical vein endothelial cells. The green we see is representing the junction of the cells, which are protein structures that hold the cells together. We also see the white portions, which represents the nucleus. To better understand this figure, let's break it into parts. Here, the first half represents the cells that did not have the SMAD6 protein. The second half did have the SMAD6 protein. And the top half had the static movement, so essentially the cells did not move. Then the lower half had the cells that had flow in it, so they were able to move. Now we're going to look at a graph that organized these images according to size. Here I labeled the color shape to match the ones on the graph. Take a second to look at them. Next, notice how the cells look wider in the red box in comparison to the others. Well, on the data, it demonstrates that the size of the junction we see has a ratio axis of about 8, in comparison to the other images that have the axis ratio at about 1. This difference is significant, therefore concluding that the cells without the SMAD6 protein and also placed under flow will most likely have distorted cell junctions. So today we can take away that immunofluorescence images can help create data which can then interpret into a conclusion. That's all for today. Thank you.